Now, of course, what we've been here, I'm going to talk about asexual and morphology of aspergillus. Now, I'm going to talk about the morphology and asexual production of penicillium. And later on, I'm going to give you the sexual life cycle of penicillium as an example of Eurasianus. Okay. Now, I'm going to start about penicillium here species. Okay. Okay. Of course, penicillium also basically, sometimes, of course, we call them the penicillium. Usually I forget that basically aspergillus, sometimes we call it the black mold. Okay. And sometimes we call the black mold break. Of course, specialist aspergillus niger, we have already in last lecture, I mentioned to you others. Penicillium normally known as green mold or blue mold. Or sometimes we call it blue mold. Depends. Of course, sometimes they are yellow, but that is the most common one, okay? And of course, if you notice sometimes, especially they grow much on the citrus fruit, as I showed you here, and their conidia gives that color of the bluish green or something like that of the penicillium. Now, as I mentioned to you, that penicillium are common and cosmopolitan as aspergillus. I mentioned to you here. The penicillium, okay, are as common and cosmopolitan. Cosmopolitan, that means distributed worldwide in different type of environment. That's we call cosmopolitan. Okay, and cosmopolitan as the aspergillus. Okay, as the aspergillus. Okay. And also the same thing I meant to mention basically that the conidia, okay, the conidia or sometimes called spore, the conidia of aspergillus, or sorry, of penicillium. Okay. Also like those of aspergillus, just simply to mention, are everywhere in the air. In the air. And also in the soil. Okay. Of course, aspergillus subspecies are economically very important because, as you know, they use basically, as you know, from aspergillus, from penicillium, we got the antibiotics. Okay, so they use many species for the antibiotics, and also, if you notice, there's also made in a lot of cheese like this Danish blue cheese. Basically, that Danish blue cheese, if you look at it, that green or blue color within the cheese, that all basically penicillium species. Okay, to mention this industrially. Okay, the penicillium as I mentioned, are important. Okay, and the making some of the cheese. as I mentioned, that if you put it down, that the Danish blue cheese, okay? And also antibiotics. Okay. The other thing I want to mention to you here, basically with the going to mention on the page here, just give me a second. Okay. As for its morphology, basically the mycelium of Aspergillus, okay, produce, okay, simple, long, erect conidiophores. Okay. Of course, basically the mycelium, of course, don't forget that I did that after vegetative growth for a period, of course, they start to produce what you call this conidiophore. And always that basically, as I mentioned to you, that the color of the mycelium without this, the conidia, basically they are white, okay, in color. And that's why basically if you look at, sometimes if you look at the petri dish, when they grow them, just you look at the edge of the colony, because, okay, if it's bluish, it looks white, like this one here, for example. If you look this one here, if you find it, yeah. Yeah. Usually, of course, it's growth here. That is the edges here, because it's a hyphae start. There is no spores here. Okay, and that's why this color, all the color you see is a result of the conidia or the spores we call it. Okay, but the real hyphae basically they are a little bit whitish. Okay, just to mention this point here. Now, as I mentioned, the mycelium produces, as a matter of fact, simple, long, erect conidiophore, which branch. Okay, about 2/3 or 2/3 of the way to the tip. Of the way to the tip. In a characteristic we call it in a characteristic, okay, symmetrical or asymmetrical. In a characteristic, symmetrical or asymmetrical. I just will show you this, just give me a second here. We call a broom-like fashion. Fashions. Okay, let's go to our talking about this one here. That means if you look to the penicillium, that is basically the conidiophore, and some of them basically, as you can see here, then they start basically a branch like this, okay? And that we call them, this is the conidiophore, and this is the branching, which is called 2/3 of the way. And the other branch, we call metula, and forget, but the last one, this also they produce the conidia, and of course, sorry, the stigmata. And this stigmata also could be one layer or two layers, it depends. And then, of course, they produce the conidia. So in the end, they get that type, what you call that pattern, which is look to you like a brush, okay? And that's we call it. Okay? symmetrical or asymmetrical fluke depend on the species. Okay. Now, the conidia, to mention to you here, in general, the conidia or the conidia 4 which is referred to, sometimes referred to as the brush because of their shape, known as the brush. And to mention the other point here, that and the multiple a branching of the conidia 4 also the same thing as you mentioned, and in a group of stigmata. Okay, of stigmata, which bear the long bear the long conidial chain. Okay, which bear the long conidial chain. Okay. The conidia itself would describe it going to be globose or what you call or ovoid. You mentioned this point here. The conidia, okay, are the globose or ovoid. And just to make sure, and under the microscope, they look like basically, or they resemble glass beads. Okay. And of course, as I mentioned, that the bluish color or sometimes some species yellow, it shows that all as a result of the color of the conidia. Okay. Now, just to mention this point here, okay, the greenish or bluish okay, greenish or bluish, okay, or yellow sometime, or yellow, conidia, which are produced okay. Of course, as you mentioned, are chiefly responsible okay, for the characteristic colony killer, of course, of various species of penicillin. Okay. Of course, basically, in both cases of aspergillus or penicillium, once the conidia disperse in the air, when they land, if there is a moisture, of course, going to germinate and to repeat the life cycle. You mentioned it, you okay? The conidia, okay, germinate easily. As I mentioned, they germinate easily into a germitube. Okay. From which, okay, mycelia develop. Okay. Uh, Now, basically, the subtitle here, I'm going to talk about the sexual life cycle or sexual reproduction in Aspergillus and Penicillium. And Penicillium species. Okay. Of course, always remember when I said life cycle, okay, and life cycle that will include both asexual and sexual. So basically, that's usually sometimes when we present the life cycle, present like this. You see that? Okay, because this basically shows you here, this part here represent, as a matter of fact, the asexual life cycle, in which you have the hyphae produce the conidia, the conidiophore, and in the end you have the conidia, and the conidia when mature, they are dispersed, in this case by the wind, okay? And when they land, for example, on any medium, they germinate, of course, but that germination always, you put in your mind, fungus need moisture. Simple as that, okay? Only we have some of the special species in which, like, I'm going to mention, like erisiph, on the plant, they can germinate even at zero humidity. That's something, but in general, always they need humidity, okay? And of course, later on, the conidia germinate into a germitube, and then you come to a mycelium, and the life cycle repeats itself. Now, in case of the sexual life cycle, now, as I mentioned, I'm going to use penicillium as an example, both. Usually what I have in case here, in this case, basically, the male gamete, as you can see here, is known as antheridium, and the female known as ascogonium, okay? And usually they are coiled around each other. Of course, these are the sexual life cycle on the species in which been observed or discovered, okay? And then what happened here, as the organism, basically, it is multinucleate, which is going to be for both. And later on, when plasmogamy and karyogamy take place, when plasmogamy takes place, then of course the nucleus from the antheridium is going to move into the nucleus of the or the cell of the ascogonium, and you end up with a structure like this. I don't know if you see it here, of course. And they're going to stay as a stage of karyogamy for a while, because as you see, this is characteristic of all the group where plasmogamy occur. The cell with two nuclei is going to last, it takes a period, not going to fuse immediately, and thus we call it dikaryommycota. And that's basically here, the ascogonium with binucleate cell. And later on, of course, what happened here, that is the stage in which it shows to you the development of the eclustothecia, okay, after, which is a row of cells occur around, okay, and of course, in the end, you can find in the eclustothecia gradually, later stage, in which you have the SI, and inside it basically then. But usually the SI develop after the plasmogamy. As I mentioned, you have dikaryotic, follow my point, and then the karyogamy when the two nuclei unite and meiosis will take place. Then the meiosis, after meiosis, is going to produce 8 ascospores. So that's basically a result of meiosis 1, okay? And meiosis 2, of course, is going to produce, one meiosis is going to divide, produce 4 cells, and later on, followed, of course, by mitosis, is going to produce the other 8 cells. You follow my own talk view. And of course, the clistothecia, as I mentioned to you, in case of Euryshevis, the body of the clistothecia is very thin and very easily broken. And when be broken, then the ascus is going to liberate, and within the ascospores is going later on going to germinate and produce the hive. So basically, that is present basically simply what you call the sexual reproduction or sexual part of the life cycle of penicillium. I'm going to put it down for you here. Did you follow this? Okay. As I mentioned to you in general, when we talk about here, the sexual production in Aspergillus and Penicillium, to mention, of course, in both species, okay, the perfect, or as a matter of fact, or sexual stage, in general, I'm going to talk about here, okay, of most species. I mentioned of most species of both Aspergillus and Penicillium is not well known. Okay, is not well known, or we call it, or not yet being discovered. However, to mention here, on those species, okay, in which sexual life cycle is known, can be summarized as follows. Okay, going to go out here. The first going to mention, of course, the sixth organ, we call it antheridia and ascogonia as for male and female respectively. Okay. The 6 organs, which is antheridia, as the male, okay, and ascogonia as the female here, okay, usually are produced close to each other. Okay. really close to each other, as I mentioned to you here. Okay. And also, and of course, both of these hyphae, don't forget, are multinucleate as a characteristic of the organism here. Both are multinucleate. Okay, both are multinucleate, and also to mention are also elongated structure, as I showed to you. Elongated structure. Okay, and sometimes they are the M3D, of course, and they get helical around the escugonia. To mention this point here. Okay, and often helical, and they coil around each other. Okay. Then the other point I'm going to mention to you, of course, then of course the nucleus the nuclei move from the antheridia to the escugonia. To mention this point here. When the nuclei from the antheridium have entered that of the escugonial has entered. Okay. Entered that of the escugonial cell. Okay. Then the two nuclei pair. Okay, they compare. Okay. Before that, then continue after this here. All right. Just to mention, of course, after the pairing of the nuclei, okay, after the pairing of the nuclei, okay, what happened here? Then the what you call the escugonium produce many hyphae. Okay, to mention here. Then the escugonium okay, it produce a number. Sorry. We call a number of escugenial hyphae. That means the hyphae they are developing from the escugonium. Okay. then sconium, sorry, mention here, produce a number of ascogonial or ascogenius hyphae, okay, which branch within the developing ascocarp. I will explain this to you. Just give me a second here. Within the developing ascocarp, all right? Ascocarp. And what happened here? And the branches are of different length. Of different lengths. Okay. And the SI okay, which are formed at their tips okay, are produced at different levels. Okay. So basically what I'm trying to show to you here that basically if you have, this is the ascogonium, because it doesn't show on that picture there. So that is the ascogonium. And when the nuclei, okay, when the two nuclei of the antheridium, they unite in pairs like this, okay, then of course, they produce, as a matter of fact, a hyphae. And basically, as you can see here, all this like that. This is within the structure is developing another hyphae, developing what you call the clustethecium around it. Another hyphae basically producing this. And that's basically each one in the end is going to have a cell basically that the SI, as a matter of fact, is going to produce here. Because remember that here, that what happened here, when these two nuclei, first of all, this is the first stage in which two nuclei come together from the antheridium to the asconium. This is plasmogamy. Okay, now and then this plasmogamy, or which we call the two nuclei, which become like a dikaryotic, they will stay for a while, and later on when chirogamy occur and meiosis occur, then that is basically these are developed and they produce, of course, the ascus there at different level. And that's basically in the end when you have the clustethecium like this with many cells like this, you can see the ascus at different levels. Okay, because from the beginning they arrange like this one. Okay, follow my point of view here. Though basically here it doesn't show the stage, but it just shows you as all in different level here. Okay, you follow my point of view that when they unite and the cell, but later on they develop hyphaes, and these hyphae with different length, and in the end you end up with the SI also. These are the SI. The sac is going to be at different levels. Okay. Okay, but of course, other hyphae, which are sterile, we call sterile hyphae, these are going to form what you call the cells, which is produce the fruiting body or the clustophysium. Okay, is that clear? Okay, this means you at different level. Now, to continue with this one here. Okay, now at early stage, the clustophysium, okay, of course, begin to develop. They begin to develop, okay, as a single layer of cells. That means this the clustophysium as one layer of cells, okay, which is not as some other species we call stroma. Stroma basically when you have the wall of a clustophysium has many layers. I'm going to show it to you later on another example, okay. And early stage the clustophysium begin to develop, okay, as sorry, this is develop here as a single layer a single layer, okay, of cells around, okay, around the sex organ, means because the sex organ already there, okay, around the sex organ as I showed you here. Around the sex organs, okay. And of course, basically, an early stage, the clustophysium here, don't forget that here. Then it mature. Okay, it matures into, later on, into a small globose circular ascocarp. Okay, ascocarp means is a term for the either called ascocarp or you called ascoma or you called fruiting body, but because circular, it is also known as the clustophysium. Is that clear or confusing for you? Please let me know. Okay, ascocarp. Okay, because the reason we use this name carp, okay, because what happened here in in the early days, in the beginning classification, they used to group the fungi, as I mentioned to you, as a plant, but we call them non-photosynthetic plant. You follow my point of view here? And in the plant, of course, when you have the flower, okay, or when you have the female ovules there in the ovary, okay, the ovary itself is called carpel, okay, and then of course what you hear, the fruit, the carpel actually called what you call carpel, and that's basically ascocarp. So basically that as comes all that terminology from there. But so much ascocarp of the fruiting body or the ascoma is the same thing here. Okay, ascop, which generally in the beginning, okay, yellow, soft, okay, and cottony. Okay. And inside, of course, the asci themselves. So basically, the body here is represent what you call, as I mentioned, you have the clustophysia, okay, which is could be one cell or two cells. Now inside it, just for your information, if this is the ascocarp, or whether you call it ascoma, or you call it ascoma, okay, or the name of it, ecdysothecia. And of course, then this is the ascus. Give me a second here. This is the ascus, and inside it are the ascospores. All right. Soft yellow, the ascocarp here. Okay. Now the asci, which is inside the asci, okay, and also are globose or circular. Globose. Okay. Now, at maturity or immaturity of the asci, in maturity, okay, the asci dissolve, okay, dissolve, leaving the ascospores free within the ascocarp. Within the ascocarp, okay, which is the same thing. The ascocarp is, or in other word here is the clistothecium. Okay. Then of course later on when the ascocarp is cracked, and then after breaking or decaying of the ascocarp, the ascospores, okay, are liberated, okay, and under favorable condition, they germinate into a new hive. Okay? So as I mentioned here, then after breaking or decaying of the ascocarp or clistothecium, the ascospores are liberated, and under a favorable condition, they germinate into a new hive or new organism. Is that clear? Okay. Now I'm going to move to another order of the, what you call, within this group of the ascomycete, which is known this order Erysipelas. Okay. So another order here known as Erysipelas. Okay. This order also, as a matter of fact, is commonly known as the The powdery mildews. Okay, erysipelas, known as the powdery mildews. The reason why, because the member of this group, they are mostly parasitic on plants, and when parasitic plant, of course, they produce what you call like white powder like this. This is the leaf like this. Did you see it? You focus on it. Okay. And of course, these are the powder. Basically, what happened here, it looks powder, but in reality, they are all either conidia or either basically the Christothecia. But this order here, of course, in the end, the plant will die, of course, if no treatment or care to it. And this order also, the sexual production produce also Christothecia. Okay, but the Christothecia in this order, they are quite very well developed. They are thick and circular, and the outer wall consists of many layers. Okay, talk about it. Okay, so basically in the beginning, let us talk a few lines on this one here. Okay, about this order, would you call it the erysipelas order? The erysipelas of Parliamento. Okay. Members of this order are parasitic on plants. Okay. And the infected ones means the infected plant, and the infected ones okay, and the infected ones, okay, produce a mealy powdery white stratum on the surface of leaves. Okay, of course, a number of plants. Okay, did you follow this? Now, the example I'm going, the two example I'm giving you, this one here, one is known as microsphere and the other one is known as erysipelas. But mostly I'm going to discuss erysipelas, okay, or microsphere. There's a big difference with this cohort. Example of the species here of this order, example, known as microsphere. Okay. Species, because there are many species of Mycology, and the other one known as erysipelas. Species, it's genus, okay. Now to talk about basically at the beginning of this group as the somatic structure or vegetative structure. Now, this species, basically the mycelium of this species called obligate parasite. Obligate parasite, that means we cannot grow the organism on a media unless it should have the host. Okay, in that case, okay, because now the mycelium, okay, the mycelium is obligately parasitic. Okay, is obligately parasitic. The mycelium is obligately parasitic on a specific host. On a specific host, because all attempts okay, to grow them for prolonged period basically in an artificial medium or culture, okay, have failed or have so far failed. Okay. Now, this organism of the erisiph, characteristic of it is like this. When they produce the, do you see here? When you have the mycelium and produce the conidifor, usually compared to the conidia is a single chain like this. Okay, and that's how it looks like. Basically, the clustothecia is quite thick, and usually the clustothecia also with they produce appendages, as you can see here, and these appendages sometimes with different structures. Okay, it depends on the species. I'm going to talk about it as I go more. Okay. Now, basically what happened here, the mycelium, okay, spreads over, okay, the mycelium spread over the leaf, okay, from original, okay, from original point of infection, okay. Give me a second here just to infection, okay. Penetrate into the host leaf, okay, and absorb nutrients by Historia. Remember Historia, we mentioned these filaments in which basically they, okay. Now, I will explain this point here to you, just give me 1 second. Now, usually the hyphal cell of RSF or microsphere, they are uninucleate. So to mention this point here, okay, the hyphal cells, okay, of most species, okay, are uninucleate. That may contain one cell. Now, as I mentioned here, when the mycelium separate over the leaf from original point of infection, and okay, now usually what happens here, the fungus like this, usually of course, if this is say a leaf, okay, say this is a leaf, cross section of a leaf, and okay, this is a leaf inside, and this is the upper epidermis. Okay. Now, when the spore land down like this, usually these fungus, they do like this, and they germinate, and when they move like this on the leaf, okay, sometimes they do structure. If I enlarge it like this, they move like this, the hyphae, and they develop something like that. You see this notch here? Okay, and then they push themselves inside until, of course, once they go inside, because they have to crack the cuticle and the epidermis to, okay, and of course they produce enzymes, okay, to dissolve it, and then of course they go inside, and of course they separate between the cell, and that's what mentioned here. The mycelium separate over the leaf from original point of infection, penetrate into the host leaf, and absorb nutrient by Historia. Historia, of course, is that the hyphae which are specialized in absorbing the nutrient. Okay. Now, of course, basically that is shown to you here. Now then, we're going to start basically with the life cycle of the organism. Okay, life cycle of microsphere, or receive the same thing of microsphere. And okay, usually we have microsphere allen in the species here. Now, then basically we'll start with the asexual reproduction. I don't think there is enough time to finish it, but I have the experience because only left about 3-4 minutes, and the screen will shut down here. Now, let me show it to you here. Now, this is the organism here. Okay. Now, this fungus, as a matter of fact, when it lie down when mature the conidia, okay, then it goes by the wind, and when it lie down, the same thing as any organism, it germinates. But as I mentioned, characteristic of this organism, they can germinate on the surface of the leaf even at 0 relative humidity. Okay. With regard to the sexual life cycle, it's very simple to that of Penicillium and Aspergillus, but of course the sex organ are different. As you can see here, you have Antheridium, and also they have Ascogonium, but they are very short and they curve around each other. Now, of course, the same stages they go, and they what call plasmogamy, karyogamy, and then, but as I told you, and this one here, the Clistothecia are quite like a stroma. A stroma where there are a layer of cells, okay, and inside it, the number of the ascus, it depends really on the species. And also this species, as I showed to you here, okay, that usually the ascoma or the Clistothecium, they have appendages, with different type of ending like this, okay? And all that basically it depends on the species, because by this way, they cling over the surface of the leaf. Because most of the time, the parasitic leaf like this, for example, I'm going to show it to you here. There's another picture here of other species here, but usually when they develop the Clistothecia, okay, if you can see this one here, they develop, and this is of the surface of the leaf, so that they kind of click on the surface of the leaf, okay? So basically, of course, what happens, the same thing, the Clistothecium in the beginning are thick, but in the end, of course, going to decay. And that is shown to you here when you have the conidia with a chain of conidiophore, one state, okay? So basically, this is the life cycle of what you call Microsphaera, or the powdery mildew, and I will give you the notes basically next lecture. Okay. Do you have any question left? Of course, your exam is going to be on next Friday, I guess, something like that. Yeah. Okay. And of course, I will send you note to clarification. Okay. And what are the material included? Hopefully by Monday or Tuesday, I will send you that email. Okay. Well, have a good day. Any questions? Well, everybody's okay, I guess. I don't know.
Thank you.
You're welcome. Take care. Bye.
Thank you, Professor.
Thank you, Professor.
Yes. Yes. How long the essay? Well, I think I mentioned to you that should be no less than, I think, 7 pages or so, or 5 pages. I sent that email. Yeah, if you want, I would send the email again for you, just to remind you again. I think that's the best way, okay? Yeah, thank you very much. Take care. Bye-bye.